Good morning. It's March 23rd, and this is your daily brief in science. Here's everything you need to know. Alectinib may boost GD2 CAR therapy in ALK-mutant neuroblastoma by lowering PD-L1 on tumor cells, potentially enhancing how CAR T cells proliferate and kill cancer. Neuroblastoma, especially ALK-mutant forms, often faces chemoresistance and an immunosuppressive microenvironment that limits immunotherapy effectiveness. In preclinical models, the combination of alectinib and DC-alloganglioside 2 CAR T cells increased CAR T proliferation, cytokine release, and tumor killing, with sustained tumor control and delayed relapse. The mechanism appears to involve downregulation of PD-L1 on neuroblastoma cells, reducing PD-1/PD-L1-mediated checkpoint signaling that typically dampens CAR T activity. By mitigating this inhibitory axis, alectinib improves CAR T cytotoxicity and persistence against tumor targets. Researchers emphasize a dual-targeted, personalized approach as a promising path for high-risk ALK-mutant neuroblastoma. The study, published in the British Journal of Cancer on March 23, 2026, provides preclinical evidence to support moving toward clinical translation with carefully designed trials. Key questions for trials will include how to optimize dosing and timing to modulate PD-L1 without increasing toxicity, and how to monitor biomarker responses during therapy. Broader implications point to a paradigm shift toward combining targeted kinase inhibitors with immunotherapy in pediatric cancers, and the approach may extend to other tumors driven by oncogenic signaling and immune suppression. The authors also discuss exploring multi-antigen or armored CAR designs and biomarker-driven strategies to refine therapy and address potential resistance. A new chapter in plant immunity emerges from wheat, where scientists have identified an octameric resistosome formed by the activated CC_G10-NLR receptor Wei3, marking the first octameric resistosome observed in plants. Published in Cell, the study positions wheat as a powerful model for immune signaling and highlights CC_G10-NLR pathways as targets for engineering durable disease resistance. The activated Y3 resistosome assembles into an 8-subunit ring that functions as a calcium channel, driving K2+ influx to trigger plant immune responses. Cryo-EM confirms the octameric structure and shows activity in plant cells but not in animal cells, implying plant-specific cofactors or membrane environments are required. The work also extends to Arabidopsis thaliana, where a homologous activated RPS2 forms an octameric resistosome and induces Sa2+ influx, suggesting conserved activation mechanisms across plant lineages. Cross-species conservation is underscored by the Arabidopsis CC_G10-NLR homolog RPS2, forming an octameric resistosome, indicating a shared architecture in both monocots and dicots. YI3's 8-subunit ring-shaped complex acts as a calcium channel to drive signaling and immune activation in plants. This octameric resistosome stands out from previously known pentameric and hexameric forms, expanding the known diversity of NLR-powered immune responses. The discovery relied on expression in Nicotiana benthamiana, followed by purification and cryo-EM, to resolve the activated resistosome structure. A gain-of-function mutation in Y3's LRR domain, derived from the spontaneous wheat autoimmune mutant M3045, drives constitutive immune activation and reveals regulatory aspects of NLR activation. A new look at nanoscale machines built from DNA is reshaping what's possible inside living systems and manufacturing. Researchers are developing DNA-based robots designed to operate at the molecular level, with aims to deliver targeted therapies and assemble nanodevices. To make these machines controllable, teams are exploring design strategies that balance stability and motion, using rigid joints, flexible components, and origami-inspired folding. The field is moving toward practical uses like nanosurgeons that identify specific cells and deliver therapies, or even capturing viruses such as SARS-CoV-2. A scholarly review grounds the work, referencing a foundational study on designer DNA machines with authors, a date, and a digital object identifier. Major challenges include unpredictable Brownian motion at small scales, scaling from macro concepts to molecules, and designs that remain static or underperform. Additional hurdles point to limited robot functionality and a lack of infrastructure, including databases on DNA mechanical properties and robust simulation tools. Advancing the field will require cross-disciplinary collaboration, standardized DNA parts libraries, AI-assisted design simulations, and new biomanufacturing methods to scale DNA robots. Future directions call for interdisciplinary innovation, with standardized libraries, AI-enabled dynamic simulations, and manufacturing advances to bring DNA robots to real-world use. Control of these robots combines biochemical methods like DNA strand displacement with external triggers such as electric, magnetic, and light fields to drive motion. DNA strand displacement provides precise programming through fuel and structural strands, enabling high-accuracy control of robotic behavior. In short, control methods blend chemistry and physics, encoding actions with DNA strand displacement, while external stimuli drive movement and shape change. The authors envision a future where these robots are biological, programmable, and intelligent, capable of mastering the molecular world. A major figure in neuroscience is stepping aside at MIT as Tsai ends a 17-year run guiding the Peikhauer Institute. Under her leadership, the institute expanded from 11 labs and about 200 people to 16 labs and around 400, and it linked directly to two MIT-wide initiatives aimed at aging and cognitive disorders. Tsai pushed significant breakthroughs in Alzheimer's research, including work on CDK5's role in neurodegeneration, epigenetic regulation, DNA damage in disease, and APOE4 pathology using human stem cells. Her translational impact includes the genus approach, gamma entrainment via sensory stimuli, that has moved toward pivotal clinical trials through an MIT spin-off. She also strengthened ties with the Peikhauer Foundation, now the Freedom Together Foundation, to fund junior faculty, private grants, and fellowships, and expanded the Institute's Innovation Fund. Two campus-wide initiatives, the Aging Brain Initiative founded in 2015 and the Alana Down Syndrome Center established in 2019, reflect Tsai's strategy to galvanize MIT's engagement with neurodegenerative and cognitive disorders. A national search committee co-led by Matthew Wilson, with input from senior colleagues like Nergis Mavalvala, is identifying candidates to succeed her as director. Tsai will focus on her own research and continue leading MIT's Aging Brain and Down Syndrome efforts through the transition, with the spring 2026 search underway and observers noting the Institute's ongoing prominence and the importance of a capable successor. A new study proposes a unifying mechanism for Alzheimer's disease by showing that amyloid beta and tau may disrupt microtubule function through competitive binding to microtubules, linking two long-standing players in the disease. Amyloid beta binds to microtubules with affinities similar to tau, and structural similarities between amyloid beta and tau's microtubule binding regions suggest overlapping binding sites on microtubules. The framework, detailed in Schoff, Derbez-Morin, Kai and Julian's 2026 paper in PNAS Nexus, offers a simple and unifying theory for the underlying cause of Alzheimer's disease. This framework could explain why amyloid plaques don't always correlate with cognitive decline and why many amyloid-focused therapies have had limited long-term success by highlighting early microtubule disruption as a critical event. If confirmed, the theory could shift therapeutic focus from merely clearing plaques to preventing amyloid beta from interfering with microtubules or enhancing neuronal clearance of amyloid beta. The study's authors emphasize that this integrated explanation connects decades of disparate findings and points to new avenues for treatment development. The proposed competition could cause the neuronal transport system to break down, with tau misbehaving and aggregating in areas outside its normal location when not bound to microtubules. Observations that lithium stabilizes microtubules and is associated with reduced Alzheimer's risk support the idea that preserving microtubule integrity could counteract amyloid beta's disruptive effects. The microtubule nexus hypothesis posits that amyloid beta's toxic effect may begin by displacing tau from microtubules, leading to tau destabilization, increased tau phosphorylation and aggregation, and impaired microtubule stability. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.